Hello, it's that time of year again. This week that I'm gonna be studying for all my exams. So this is gonna be my genetics lecture stream, like the full one, because I had like an intro one where I talk about like the first half of the unit, and I'm gonna talk about the second half. So I basically compiled everything into one. So first I'm gonna do this genetics stream, and then I'm going to do a stream where I work all the problems, and hopefully I can squeeze all of it into because I really have only an hour and 15 minutes to do this. So let's go. Okay, so genetics is the study of heredity. It's the study of heredity and expression. So heredity is basically the transmission of genes from one organism to another through like reproduction and whatnot. And expression is the genetic information transferred into a genotype. So basically, like the genes expressed as a trait. So there's a couple of theories as to heredity. So the first is spontaneous generation. An example of this would be religion, or also the swan egg experiment, where organisms started to grow out of a completely sterile environment. So this is like life happening from like just out of the blue, like nothing came from it, you know? Preformationism is the idea that we evolved from many miniature versions of ourselves. For example, sorry, I'm stuttering. The homunculus, just like a sperm human. So we start as like tiny versions and then we grow older. So it's kind of like a baby, except even smaller version of that. So pangenesis, or gemmules, is the idea that throughout our lifetime, pieces and bits of ourselves, like a small piece of our hair, small piece of our eyes, all the organs shed tiny bits called gemmules, and they gather in our reproductive organs, and that is passed on through reproduction. So pangenesis follows the idea of blended inheritance, which is random, like random assignment, and like the offspring has characteristics of both parents randomly, and then also acquired characteristics. So characteristics is like characteristics that an individual has in a lifetime will be passed on to the offspring. So, for example, if I cut off my arm or whatever, like if I lose my arm, then my offspring might not have an arm, I guess. Like just things that you develop in your lifetime will be passed on to your offspring. So, epigenesis, this is pretty much, this proves everything else. This is the idea that babies are made from information and not physical characteristics. So, like, pangenesis is like physical characteristics being passed on. However, epigenesis is like an embryo is formed from information, genetic information that has nothing to do with like actual physical characteristics unless the environment persists or allows for it to. So, what is life? Life is the ability to grow, adapt, and react to stimuli. The elements of life are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And the macromolecules of life is carbohydrates, lipids, and nucleic acids. Genes are the instruction of making proteins and DNA. And basically, genetics is just the discussion of DNA. So we're going to get into DNA. So DNA discoveries. So through a whole bunch of scientists and discoveries, we were able to find out that proteins coagulate under heat, and there is the nuclein, which is a protein. But the nuclein has a non-protein part called nucleic acid made up of nucleotide sequences, and nucleotides are made of sugar, phosphate, and a nitrogenous base. There is a transformative principle we found out about DNA, which is through smooth and rough stains of, I think, a virus. So basically, the smooth strain was virulent and the rough strain is virulent. If you treated that smooth strain with heat, it would have been avirulent. However, if you put a smooth heat treated smooth uh, strain, sorry, with a rough strain, the smooth strain becomes virulent again, even though it was treated with heat. But yeah, so there's a transformative principle there. So we were able to find out DNA is the hereditary material. Allows for research, pharmacogenetics, pharmacology, agriculture, DNA libraries, and diagnostics. And the tetranucleotide hypothesis is basically that like the nucleotide has 4 types of bases: adenine, tyrosine, or thymine. I actually don't know if it's thymine. Here, let me see. It's okay, yeah. So it's adenine, thymine, cytosine, and guanine. I don't know what tyrosine is. I don't know why I said that. Oh, it's an amino acid. It's not anything. Okay, but anyways, we were there's a scientist called Chargas. He has 2 Chargas rules. So the first one is ratios. So the ratio of A and T is going to be similar in all organisms. Sorry, in all organisms, and the ratio of C and G are going to be the same. So there's an equal amount of A and T, and then there's an equal amount of C and G. And then the second one is variation. So the second Chargaff rule is basically that the number and amount of base pairs and bases will vary throughout organisms. The image B51 that was pretty much discovered by Rosalind Franklin helped identify the structure of a DNA. So there's DNA parts. So a nucleoside versus a nucleotide. So a nucleoside has only 2 parts. It only has a base and a sugar. However, a nucleotide will have the base, the sugar, and phosphate. So that's the difference between a nucleoside and nucleotide. And they have 4 bases: A, T, C, and G. A and G are purines, which are double ringed, and then C and T are pyrimidines, which are singular ringed. They have 3 types of bonds. There's a phosphodiester bond, which is the backbone of DNA and links sugars, like the 5' carbon sugar of one sugar to the 3' carbon of another sugar. The second was an N-glycobond. This links the base to the sugar, and this can be in a syn or anti configuration with the sugar and the base being towards each other or away from each other. The hydrophobic interactions of DNA will allow for the twisting of DNA, so it'll have the bases go towards each other like this, kind of. Like think of this as the backbone, and this is the my 2 things are the bases. So it'll look like this. So they'll be like this stacked on top of each other. However, since the bases kind of hate each other, it'll cause twisting per like, you know. Okay, so there's 3 types of DNA. There's A, B, and Z DNA. A DNA is dehydrated, right-hand formation, tilted, anti-configuration, wide and short. B DNA is hydrated, right-hand formation, stacked bases, anti-bases, and this is the basic type of DNA. And Z DNA is left-hand formation, zigzagged, alternate bases, syn and anti-configurations with purine, pyrimidine, CG patterns, a lot of those repeating. So again, A DNA is kind of big and fat. The B DNA is like the basic DNA that you'll see in any picture, and Z DNA is kind of like frizzy, like, you know. Anyways, so there are a couple of discoveries that we're going to discuss. Other than the discoveries up here, we have Mendel's discoveries from his pea experiments. So he determined that you needed at least 2 alleles for one phenotype, and then there for a recessive trait like a Mendelian inheritance, a simple dominant trait, the recessive allele in the parent will disappear in the F1 generation. However, it's going to reappear in the F2 generation in a 3-to-1 ratio. And then there is a multiplication and addition rule. So if you see the word and in the equation or in the problem, you need to multiply. If you see or, you need to add. So this has to do with like probabilities. So it's like what is the probability of a individual having black hair and brown eyes, you would multiply. And if it's black hair or brown eyes, then you would add each probability. Okay, so there's 3 laws. The first law is allele segregation. So this is has something to do with diploid cells and random assignment. The second law of independent assortment has something to do with like not being dependent on each other, like the alleles will not be dependent and they will completely separate and assort themselves in a completely random and independent way. The third law of dominance explains trait masking. So this is like for dominant and recessive traits. And then there's a chromosome theory of inheritance, which basically explains chromosome combinations as the primary mechanism of heredity. Okay, we're going to move on to proposed mechanisms of DNA replication. So this is going to be about the cell cycle and DNA replications. We first talked about genetics as a whole, and then we talked about discoveries of DNA and types of DNA and just DNA. And then now we're going to talk about DNA replication in the cell cycle. So we have the first model, which is the model that we follow today. It's called the semi-conservative model. This is when the created DNA that is replicated has one parent and one daughter strand. The conservative mechanism means that the parent strand is completely conserved, and then the daughter strand is all original. Like there's all new nucleotides. There's nothing retained from the parent. And then dispersive model is everything is interspersed between parent and daughter. So like everything is mixed up, not like nothing's conserved at all. Anyways, so the DNA replication, there are a couple of there's a couple of amino acid ? No, there's a lot of proteins and enzymes to know, and also a couple of phases. So initiation is the first phase. This happens on the replicon. The replicon is just basically a segment of DNA that the replication will start on. The initiator of proteins are going to be the first types of enzyme that are going to go to that replicon, and it'll bind to the origin of replication. So they will choose an origin of replication to bind on that replicon. Okay. Second of all, we're going to unwind. This is the phase of unwinding. Helicase is going to break a bond on that side of origin of replication, and often this is origin of replication, and the bond is going to be broken near an AT sequence or AT bond, because those only have 2 hydrogen bonds, and I believe that CNG only it has 3 hydrogen bonds, so it's harder to break. So yeah, the helicase is most likely to break something where there's a lot of A and T sequences, so it's just a lot easier to do all this. And then, so that's the function of helicase, to break the bond. Topoisomerase or gyrase will reduce the torsional strain between bases. So this will allow the helix to unwind. And then the single strand DNA binding proteins will help stabilize the DNA until elongation. This all will create a replication bubble in a fork. So you'll see right here, this is the replication bubble, and then this is the replication fork that will separate this double-strand DNA to the single-strand DNA. So the third part of this will be priming. So primase will add primers to the DNA. So they'll add some primers right here, a couple of primers right here. And these primers basically allow DNA polymerase to add on to the new daughter strand. Will add nucleotides. So the fourth part of this will be elongation. Again, I just said DNA polymerase is going to add nucleotides to the primers to make the daughter strand. So the polymerase E, I guess, will create the leading strand continuous from 5 to 3. So this will be this top one. It'll move from the bottom of right here. It'll be the 5 prime. And it'll move in towards the replication fork continuously to the 3 prime end of that, the bottom of that unzipped leading strand, or the unzipped DNA. And then the polymerase B will synthesize the lagging strand discontinuously from 5 to 3 again, but starting from the replication fork and forwards in Okazaki fragments. And the reason for this is it doesn't know where to stop, until it stops codon, I think. I don't know. I don't really know when it stops, but all I know is that the leading strand is going to stop whenever the replication fork stops, and then the lagging strand is just going to lag for a little bit. So the primers after this will be removed, and ligase will seal those gaps between the Okazaki fragments. And telomerase, this will add to the 3 prime end of the DNA to prevent gene deletion and DNA shortening. So basically, DNA is going to be compiled into ends of chromosomes called telomeres, and telomerase will keep adding on and elongating the DNA, causing repetition sequences, so the DNA won't become short and we won't like it prevents aging as well. So polymerase, there's a couple of notes to note about DNA replication. Polymerase needs a primer to add nucleotides onto that the synthesized strand, because if it doesn't have that, it really can't do anything. So basically, primase is the initiator, and then polymerase will finish, or at least continue what was started, and then the entire process of replication is semi-conservative, like we said before. And then it's high fidelity, which is really, really accurate. I believe it's one mistake per 1 billion nucleotides made. I'm not really sure, but all I know is high fidelity, high accuracy. So replication is bidirectional, meaning that leading strand will go this way from, I guess, right to left, and the lagging strand will go left to right. However, it is only in one direction. So kind of confusing, but it's bidirection, meaning it goes from like right to left and then left to right. However, it only goes in the 5' to 3' direction. So it makes sense because it's opposite. The leading strand starts from the 5' and goes to the 3' end. The 3' end will be on the double strand DNA, and then the lagging strand, the 5' end, will be on the double strand DNA, and then the 3' end will be on the single strand DNA, if that makes sense. Okay, so we're going to talk about the cell cycle now. It starts with GO, which is resting phase, G1, which is growth, S, which is replication, then G2, which is growth again, and then M, which is mitosis, which is division. So mitosis produces 2 identical cells and only happens in somatic cells, and meiosis is different because it produces 4 haploids, gametes, and this happens in germ cells only. So what meiosis is, it has 2 phases. So meiosis starts with duplication, and then P1, which is prophase 1. These produces 2 homologous chromosomes, or just like homologous pairs. And then metaphase is when they line up, anaphase is when they separate, the homologous chromosomes separate. And then telophase, which produces 2 haploid daughter cells. And these are identical, by the way. And then meiosis 2 will have prophase 2, metaphase 2, anaphase 2, and these involve sister chromatids that separate. Because meiosis has mitosis has identical daughter cells, but meiosis always produces haploid cells. I don't think though that the first meiosis are identical though. But anyways, so meiosis 2 will involve sister chromatids, and they will separate at anaphase. And then telophase will produce 4 haploid gamete results. So the purpose of meiosis and all of this is to maintain the number of chromosomes in individuals as compared to mitosis, which do not have the number of cells and like chromosomes and whatever. And the second purpose is for genetic variation, which results from recombination of genes. Homologous chromosomes are just basically similar chromosomes that have the same genetic info at the same loci. So like they have the same genetic information at like same kind of like locations, but they're not always identical, but they can be. So to calculate the number of gametes, it's 2 to the power of n, when n is the number of heterozygotes. So for example, if it's capital A, lowercase capital B, capital C, lowercase c, then the number of gametes possible would be 2 to the power of 3 because there's 3 heterozygotes. If it's capital A, capital B, capital C, lowercase c, then it's only 2 to the power of 2. So one cell will have 23 chromosomes, meaning it'll be 2 to the power of 23, which means there's 8 million gametes possible. So fertilization is when 2 gametes join to form a zygote. So this means it's 2 to the power of 23 times 2 to the power of 23, which means there's 70 trillion zygotes possible. Isn't that crazy? It's so crazy. Anyways, gametes fuse to form zygotes, like I just said. There is two differences. Well, not two differences, but there's two types of replication, and you need to know the difference. So for prokaryotic cells, the DNA is circular, and it comes from only one origin, and it kind of like runs free in the cytoplasm. And it is circular. And then for eukaryotic, it's multiple linear DNA, and it comes from multiple origins, and it's tightly packed on the telomeres at the chromosome ends, like I just mentioned. And moving on from all this, we're going to talk about allelic interactions, which basically. Oh, actually, let me go over the cross types first. So the cross types first, there's a true/pure cross, which means like it's like all like like pure. So like all dominant traits are all recessive traits. So that would be if you cross an AA organism with another AA organism. Sorry, when I say AA, I mean like capital AA, capital AA, or lowercase AA, X lowercase AA. So it'll be these first two. So the a capital AA, X capital AA will be a pure capital A cross, and this will result in only capital A genotypes and phenotypes. So this would be the dominant trait in all of the lineage. So this is a complete pure, completely pure dominant. And then a completely pure recessive would be lowercase a, X, lowercase a, which is be a pure lowercase a cross. And this will result in only lowercase a genotypes and phenotypes. So a reciprocal cross is a cross where you do a cross with, like, for example, if you're going to test for one trait, you're going to test with the male having that trait and the female being a wild type. And then you're going to do another cross where the male is wild type and then the female has the trait that you're trying to test. So the purpose of that is to see if the trait is sex linked. And then there is a self cross or a test cross. So they're not the same thing, but the self cross is if you cross some like a genotype with the same exact genotype to see what it is. So for example, like an F cross where it'll be a heterozygous zygote crossed with another heterozygote. So this is called an F cross. It'll result in a 1 to 2 to 1 genotypic ratio, but a 3 to 1 phenotypic ratio. And then a dihybrid or heterozygous, a test cross will be a completely different genotype. So for example, the parental P1 cross will be 2 capital A's with 2 lowercase a's, and this will all result in a heterozygous genotypic and phenotypic ratio, or just all genotypes and phenotypes of the heterozygote. But however, like that's not the only test cross. This is just an example. But yeah, and then you have the dihybrid or heterozygous, the dihybrid that are heterozygous cross. So there's two of these that you need to know. So if it's capital A, lowercase b times all capitals or all lowercase, this will result in a 1 to 1 to 1 to 1 ratio. And then if it's like all heterozygous cross with all heterozygous, this will be a 9 to 3 to 3 to 1 ratio. So when I talk about the big six, we already talked about the first four big six. So there's a pure A, a pure capital A, pure lowercase a parental, an F cross, and then the last two we're going to talk about is a recessive back cross and then a dominant back cross. So the first one is a capital A, lowercase a by lowercase a. This is a recessive back cross because there's 3 recessive like alleles, I guess. And this will result in a 1 to 1 ratio of capital A, lowercase a to lowercase a genotype and phenotype. So a dominant back cross would be a capital A by a capital A, lowercase a. And this will result in a 1 to 1 ratio again of capital A to capital A, lowercase a. And this will just result in that 1-to-1 genotypic ratio. However, the phenotypes will just gonna be all dominant because again, it's a dominant backcross, so there's like the results are all going to present as a dominant. Okay, so last thing we're gonna talk about, or second to last thing, we're gonna talk about allelic interactions, and then we're gonna talk about multiple alleles, and then we'll be completely done, and then I'm going to move on to the problems. So Mendelian inheritance is the first type of allelic interactions. So this is the very basic type of like heredity or whatever, of inheritance. I don't know why I said heredity. So this involves like dominant and recessive genes, 2 alleles for each phenotype. And then this follows all 3 laws: the first law of segregation, the second law of independent assortment, and then the third law of dominance. So again, this has trait masking and everything. But yeah, the second type is incomplete dominance. This will result in a mixed phenotype for the heterozygote, meaning like if you cross a red flower with a white flower and you get a pink flower, that is incomplete dominance. So codominance will result in both phenotypes expressed for the heterozygote, meaning if you cross a red flower with a white flower and then you get a red and white flower, not a pink one, just a red and white flower, like for example, like red with white spots or white with red spots, this would be called codominance. Pleiotropy means one gene can affect many traits. So for example, Marfan syndrome, if you have that one gene from Marfan syndrome, it can affect your whole body. Like you can have, it basically results in like long limbs. So I guess it could affect like your legs, your arms, your torso, your neck, all of that. Antagonistic pleiotropy is one gene affecting many different like traits, but in a competitive way. So for example, the p53 tumor suppressor, it will like stop cancer cells from dividing, which is good, but it also stops stem cells from dividing, which is bad because you want stem cells to divide to help you heal and regenerate your body and all that. So that's an example of antagonistic pleiotropy. So now we have lethal alleles. There's a recessive lethal and a dominant lethal. So what a recessive lethal allele is like death. But let me show you. I'm like being really weird right now. Okay. So let me show you on the bottom right here. So we have this. Okay, this is really ugly compared to what I have written up here, but it's okay. So what a recessive lethal allele is means this is going to be the allele that is recessive, right? So if you get this trait, or if you get this genotype, it's going to be recessive, meaning either like you're going to die, basically, or it's going to be lethal, meaning you're going to die. However, if you have this, well, of course you don't have the allele, but I'm saying if you have this one, you're going to stay alive. And if you have this one, even you're going to be a carrier, but you're going to still stay alive. So for example, if this person will mate with this person, they can have a kid that has that lethal allele. However, these two people are only going to be carriers. However, with the case of dominant alleles, if you have this genotype, you will survive. However, if you have any of these, it's lethal. So that's what a dominant lethal one is, and a recessive lethal. So why do lethal alleles exist if like you're gonna die so much? So there's a couple of reasons why lethal alleles will exist, because of 3 reasons. The first reason would be genetic cheating, meaning like a lot of phenotypes, they will actually like be delayed from expression by those genes. For example, Huntington's disease, it can reduce your lifespan. However, it's not, like the onset is super duper slow. So for example, the onset is going to start happening when you're like 40, for example, or 30. So you wouldn't know that. Like you wouldn't know that like it's a lethal allele. Like some lethal alleles, you'll die from like the womb. Like, it's just gonna result in a terminal birth or whatever. But I'm saying like some lethal alleles like Huntington's will cheat, meaning they will delay the onset of expression, and that will allow the genes to be maintained in the population and actually passed on. A little small fun fact about Huntington's: the expression is based on the CAG glutamine repeats. So if there's like more than 26 of them on the HTT protein, then it'll express. But I believe like early on in your life, that won't even happen. But anyways, the second reason is that they persist in a permissive environment. So if the environment like lets them, they'll actually keep going. For example, the T4 bacteriophage on the E. coli strands. So T4 will choose to live on the B. E. coli strand, not the K12 strand. And that's how it'll like keep staying alive. And then this allows for a lot of research on indivisible genes, triplet genetic code, and nonsense codon. So that's pretty important. T4 on B. E. coli. And then penetrance is the third reason. So penetrance is basically the amount of carriers versus the amount of expressors. So like what genes, like what people will actually express the gene, and what people are just only carriers, like they just got lucky and like didn't get lethal. So for example of this would be the BRCA1 and the BRCA2 gene mutations. 80% of these people who have BRCA1 or BRCA2 mutation will actually get breast cancer, and 20% of them, of the people who have that mutation, won't have breast cancer. So just to keep in mind, penetrance will only describe the degree of expression of a trait and doesn't actually describe like if this trait will be inherited, like will be expressed or not. That's a different type of like topic to discuss for a different day. So the last thing we're going to talk about is multiple alleles, which means there's more than two or more alleles for one phenotype. So a big example of this will be blood type. So blood type is defined by three alleles: IA, IB, and lowercase I. So IA and IB are codominant, and then both are dominant to little I, which is a recessive trait. So IA or IA little I will be type A. Same goes for type B. IB or IB little I will be type B. IAIB will be type AB. Again, this is codominant to each other. So this is also the universal recipient of blood. And then type lowercase I will be type O, which is the universal donor. So fun thing to say, I guess, type A has A antigens and B antibodies. Type B has B antigens and A antibodies. Type AB has A and B antigens and no antibodies, which is why they're the universal recipient, because the antibodies won't be attacking anybody. And then type O is the universal donor because they actually have no antigens, but they do have A and B antibodies. So they can donate because nobody's going to attack them because they don't have any antigens. So antibodies basically are the attackers and antigens are the ones being attacked, if that makes sense. That's a really simple way of saying it, but for now, just think of it that way. And then we have the rhesus factor, which is another type of antigen. So rhesus, because they were first identified in rhesus monkeys. So RH plus is dominant to the negative rhesus factor. And the ratio is RH plus and then underscore 2 RH minus, because it's basically only two phenotypes, which is RH plus or RH minus. No matter whatever the second allele is, it'll be RH plus on the side, RH minus on the side. Okay, so for A, it's IA RH plus blank, or IA lowercase I RH plus blank. That will all equal A plus. And this is the same for B type blood. This is a bit confusing, I'm sorry. But I'm gonna bring it closer so you can see here. Y'all, right here. Okay. Yeah, so that's the same for B, and then for the negative, like A negative and B negative is the same thing. It's the same thing as up here. However, you see right here, we have this blank for whatever that second one is. It could be homozygous positive or heterozygous positive. It'll be positive either way. And then right here, it'll be the negative recessive right here. And copied from up here. So that's for A negative and B negative. So this is A positive, B positive, A positive, A negative, B negative. And then same thing for AB, except it's just going to be 1. So IA, IB, RH plus blank is AB plus, and then IA, IB, RH negative is AB negative. And then same here, baby eyes with RH plus blank is O plus, baby eyes with RH negative is O negative. We have a third type of surface antigens, just like the ones in the just like the rhesus vector and the blood type antigens. We have the M and N surface antigens. So it'll be like the allele will be described as LM and then LN. These are both codominant, just like the blood type. So again, right here, if you have type A, it'll be ALM with a LM. It'll be ALM. If you have type B with an N, it'll be BLN. If you have type AB with MN, it'll be ABLMN. So yeah, that is basically all. I'm going to now stop the stream and start streaming about the problems that I have worked on yesterday, and it'll help explain some of the problems, some of the math problems. So yeah, bye.